Welcome to Vet Neurojar, the podcast dedicated to exploring the intricate world of veterinary neurology. I'm your host, and today we're diving into a fascinating and rare case, meningiomas found in the fourth ventricle of cats. This is based on a recent study that sheds light on a previously undocumented occurrence in feline medicine. Would you be able to give us some background on meningiomas in cats?
Meningiomas, as many of you know, are the most common type of central nervous system tumor diagnosed in cats. They can account for a significant percentage of intracranial and primary brain neoplasms in our feline companions. Typically, these tumors are found on the surface of the brain, particularly in the area above the tentorium cerebelli, and they originate from the arachnoid cells that cover the arachnoid villi of the meninges. These tumors often compress the surrounding brain tissue, but rarely invade it. In some instances, meningiomas can arise from the third ventricle, likely originating from the tila choroidea. Now, what makes this study so unique is that it details two cases where meningiomas were found in the fourth ventricle of cats. To the best of the researchers' knowledge, this is the first documented occurrence of such cases. While meningiomas have been observed in the fourth ventricle in humans and dogs, a comprehensive search of medical literature databases revealed no prior reports of this in cats. Can you delve into how these cases were discovered?
Researchers meticulously searched through autopsy archives from veterinary pathology departments in Brazil and the United States, spanning from January of 2007 to May of 2024. They used specific keywords such as feline, cat, meningioma, and fourth ventricle to identify relevant cases. After reviewing clinical forms and autopsy reports, they narrowed it down to cases with available formalin-fixed paraffin-embedded tissue blocks for further analysis. Ultimately, 2 cases met the criteria and were included in the study. Both cats were 15 years old. One was a castrated male, and the other was a spayed female. In one case, the cat was found deceased at home without any prior clinical signs, and the cause of death remained undetermined. The other cat, however, presented progressive neurological signs over a 3-month period, including an absent menace response, which is the lack of blinking when an object is moved quickly towards the eye, head tilt, and ataxia or impaired balance and coordination. Given the poor prognosis associated with these progressive signs, the second cat was eventually euthanized. During the autopsies, some common gross changes were observed in both cats. These included well-defined, spherical to elongated masses within the fourth ventricle. The masses varied in color from white to red and had a firm texture. The expansion of the fourth ventricle led to compression of vital structures such as the cerebellum, midbrain, pons, and medulla oblongata. Based on these observations, the primary differential diagnosis considered was ependymoma, another type of tumor that can occur in the fourth ventricle. To further investigate, imprint smear samples were taken from the ventricular mass of the first cat during autopsy and stained for cytologic examination. Microscopic analysis revealed highly cellular smears with individual and aggregated spindle-shaped cells along the margins of branching capillaries, all against a background of blood. The cells exhibited wispy borders, a moderate amount of light blue cytoplasm often containing clear vacuoles and fine, elongated processes. The nuclei were single, ranging from round to oval in shape, with finely stippled chromatin and one nucleolus. There was noticeable variation in cell and nuclear size, frequent karyomegaly, which is abnormally large nuclei, and occasional binucleate cells. Notably, no mitotic figures were identified. Based on these cytologic findings, the initial interpretation leaned towards a spindle cell neoplasm, possibly originating within the brain. Histological evaluation of tissue sections stained with hematoxylin and eosin provided further clarity. In the first case, the tumor was classified as a World Health Organization Grade 1 fibrous meningioma. This meant that the tumor was composed of interweaving bundles and fascicles of elongated meningothelial cells supported by a delicate fibrovascular stroma. The neoplastic cells contained a small amount of eosinophilic cytoplasm and oval nuclei with finely stippled chromatin and one nucleolus. Moderate pleomorphism, or variation in cell shape and size, was observed, with 2 mitoses identified within a specific area. In the second case, the tumor was identified as a World Health Organization Grade 1 transitional meningioma. This tumor consisted of closely positioned lobules of meningothelial cells and fascicles of elongate cells supported by a fine fibrovascular stroma. Neoplastic meningothelial cells frequently formed scattered concentric whorls. The cells had moderate amounts of eosinophilic polygonal cytoplasm and round to oval nuclei with finely stippled to vesicular chromatin and one nucleolus. Minimal pleomorphism was noted, and no mitoses were observed. In both cases, the meningothelial neoplastic proliferation exhibited a non-infiltrative nature, instead compressing the surrounding neuroparenchyma near the fourth ventricle. This compression affected areas such as the midbrain, cerebellum, pons, and medulla oblongata. Additionally, extensive hemorrhage was observed within the tumor and in the mesencephalic aqueduct of the first case. To further characterize these tumors, immunohistochemistry was performed for ecadherin, Vimentin, and pancytokeratin AE1/AE3. Both cases showed diffuse, robust, membranous, and cytoplasmic immunolabeling for ecadherin, as well as cytoplasmic immunolabeling for Vimentin. In the second case, multifocal, weak cytoplasmic immunolabeling for pancytokeratin was observed, particularly in areas with concentric whorls. However, no pancytokeratin immunolabeling was seen in the first case. These immunohistochemical findings further supported the diagnosis of meningioma. Interestingly, one of the cats was also diagnosed with concurrent chronic kidney disease. The second cat had several concurrent conditions, including thyroid hyperplasia, cardiac hypertrophy, chronic kidney disease, and parathyroid hyperplasia. Now, let's discuss the potential origin of these intraventricular meningiomas. It is believed that they arise from the tela choroidea, a fibrovascular tissue structure that gives rise to the choroid plexus. In humans, intraventricular meningiomas are relatively uncommon and typically found in the lateral ventricles, followed by the fourth and third ventricles. This distribution is thought to correlate with the extent of the choroid plexus within these ventricles. In domestic animals, the choroid plexus tissue is less prominent in the lateral and third ventricles, but is more robust and lobulated in the fourth ventricle. While most feline intraventricular meningiomas originate from the third ventricle, intraventricular meningiomas have been reported in dogs in both the lateral and fourth ventricles. Given the rarity of intraventricular meningiomas in veterinary medicine, the precise frequency of tumors within specific ventricles remains uncertain. The clinical presentation of cats with meningiomas often involves insidious and progressive neurological deficits that reflect the tumor's location. In this study, one cat showed no clinical signs, while the other exhibited clinical signs consistent with lesions in the brainstem areas compressed by the tumor. Due to the slow-growing nature and intraventricular location of these tumors, clinically silent cases may occur until the tumors reach a considerable size. Determining the precise location of intracranial tumors based solely on neurological examination can be challenging due to secondary effects of the mass. In human patients with meningiomas in the fourth ventricle, clinical signs often relate to internal hydrocephalus and intracranial hypertension resulting from obstruction of cerebrospinal fluid flow. However, neither of the cats in this study showed evidence of hydrocephalus. Intraoperative cytology can play a crucial role in providing an accurate and rapid diagnosis of feline central nervous system tumors. Cytologic findings of meningiomas can vary, reflecting their morphology and cellular arrangement. For instance, epithelioid cells may form clusters in the meningothelial subtype, while elongate cells form fascicles in the fibrous subtype. The transitional subtype may exhibit both meningothelial and fibrous patterns. Despite these variations, certain features such as whorls, psammoma bodies, and intranuclear cytoplasmic invaginations can aid in cytologic recognition during routine evaluation. Although these distinctive features were not observed in the autopsy smears from the mass in the fourth ventricle of the first case, fibrous meningioma was considered as a differential diagnosis due to the presence of neoplastic elongate cells. Meningiomas can exhibit a wide range of histologic variations, reflecting the mesodermal and neural crest origin of arachnoid cells. In humans, intraventricular meningiomas are predominantly fibrous tumors. Data from large case series of feline meningiomas indicate that most cases are World Health Organization Grade 1 transitional meningiomas, followed by fibrous, meningothelial, and psammomatous tumors. Rare subtypes include angiomatous, papillary, and anaplastic meningiomas. However, given that only 2 cases of feline fourth ventricle meningiomas have been reported, it remains unknown whether these tumors tend to be of a particular World Health Organization grade or subtype. Several markers, including Vimentin, pancytokeratin, ecadherin, CD34, claudin-1, glial fibrillary acidic protein, glucose transporter 1, laminin, encadherin, and S100, can be useful in supporting the diagnosis of intraventricular meningioma. However, immunolabeling may vary significantly among tumors, and no validated immunohistochemistry panel has been standardized to confirm the diagnosis. In the cases described in this study, positivity for Vimentin and ecadherin supported the diagnosis of meningioma. This study highlights the importance of considering meningiomas as a differential diagnosis for tumors of the fourth ventricle in cats. Although rare, these cases expand our understanding of the diverse ways in which tumors can manifest in our feline patients. Further research and case studies are needed to fully characterize the clinical behavior, pathogenesis, and optimal treatment strategies for meningiomas in this unusual location. Thank you for joining us on Vet Neurojar. Keep those minds inspired, hearts light, and tails wagging.